All rights reserved. We reserve all rights. A Farewell to Virology Expert Edition was published by Dr. Mark Bailey in 2022. It is an as yet uncontested technical paper, and this film series is designed to make it easier to understand why. The full 29,000-word essay can be downloaded for free at drsambailey.com. Virology invented the fictional theoretical virus model, but has consistently failed to fulfill its own requirements to prove their existence. It is claimed that viruses cause disease after transmitting between hosts such as humans, and yet, outside of cartoons, computer models, and Hollywood movies, the scientific evidence for these claims is missing. One of virology's greatest failures has been the inability to obtain any viral particles directly from the tissues or fluids of organisms like humans said to have viral diseases. In order to obfuscate this state of affairs, virologists have resorted to creating their own pseudo-scientific methods to replace the long-standing scientific method, like rigorous control experiments, to try and disprove their original hypothesis. But virologists have also nefariously changed the dictionary meaning of words in order to mislead the public and support their anti-scientific practices. For instance, to isolate means to cause to be alone or apart from everything else. Yet virologists have never isolated this extracellular vesicle particle alone and apart from the thousands of other particles found in human tissue, fluids, and their petri dish and test tubes full of monkey kidney and fetal bovine calf tissue, to genetically characterize it and prove it even is a virus particle, and not just the usual, normal, regular dead cell tissue debris breakdown particles from dying, rotting cells in their petri dishes. You must isolate that particle alone and apart from all of that other stuff to prove it is a virus and not just cellular debris garbage.
What do we mean by virus isolation or virus purification? These are jargon words in virology, and they're not very precise. They mean different things to different people.
No, it specifically means to remove that particle alone and apart from all of the other identical cellular garbage decay and genetically and chemically characterize it to prove it is a unique thing called a virus and not just more cellular garbage.
Isolation, what was it?
Isolation and purification.
Of the virus?
Yes.
Well, you isolate a virus by finding the virus which causes a disease. And you purify a virus by making just by purifying it so you get a pure virus.
Okay.
I don't understand what the issue.
They interchanged the two, and I wasn't sure
I see.
If it was the same thing or if it was two totally different.
No, it depends on how they used it.
Okay. Can you explain the process of HIV isolation?
Well, didn't Dr. Gallo do that? I mean, he actually isolated it. So,
Yeah.
I mean, why should I do all of this? This is all textbook stuff you're asking me.
I'm not quite sure what's behind your question about isolation.
I don't want to be your textbook. You know?
Okay.
I got other things to do.
A viral particle must fulfill defined physical and biological properties, including being a replication-competent intracellular parasite, meaning it results in identical copies of itself inside a host organism, and also capable of causing disease in a host, such as a human. However, viruses such as SARS-CoV-2 are nothing more than phantom constructs, existing only in imaginations and computer simulations. In this paradigm, cases of invented diseases like COVID-19 are nothing more than the detection of selected, tiny common genetic sequences and proteins purported, or rather pretended, to be viral. The existence of an actual virus is not required in this fallacy loop of circular reasoning, and thus, entire fictional pandemics can be built upon fictional digital creations and then falsely sustained through meaningless in vitro test tube molecular reactions. In Part 1, we'll outline some of the history of virology and its blatant pseudoscience-like failure to follow the scientific method. In Part 2, we'll examine the fraud and deception used to propagate the COVID-19 plandemic. Finally, in Part 3, we'll analyze how health institutions, mainstream media, and governments maintain the virus illusion in tandem through information control and carefully crafted narratives that parrot virology's baseless claims.
Here's the fact which every virologist agrees to, and I can even explain that a little bit. There is not one published scientific paper where any particle that you could call a virus, and remember, a virus is a thing, it's a particle, we can sort of see it under an electron microscope, even though that's problematic. But so this thing, they tell us, there's not one published paper in the scientific literature finding this thing in any biological fluid of any sick human or animal. Anywhere. And everybody agrees. All the virologists agree. They say that's not how you find a virus.
As Dr. Tom Cowan also points out in Settling the Virus Debate, identical particles that have been successfully isolated through purification, like exosomes, extracellular vesicles, apoptotic bodies, and microvesicles, have not been shown to be replication competent, infectious, and disease-causing. Hence, they cannot be said to be viruses. Additionally, the proffered evidence of viruses, through constructing and assembling alleged virus genomes like puzzle pieces and animal experiments, derives from questionable to flawed methodologies with insufficient experimental controls. As of January 2023, following extensive inquiries through Freedom of Information requests coordinated by Christine Massey, not even a single one out of 211 government health or science institutions in over 35 countries have been able to provide direct evidence of the alleged SARS-CoV-2 virus. The institutions were asked to produce any documents demonstrating the purification of SARS-CoV-2 said to have caused disease in humans via maceration of the fluid from a human said to be sick with a virus, the filtration of that fluid, and finally the use of an ultracentrifuge to find that virus, also referred to at times by some people as isolation. On many occasions, following an admission that no such evidence is held, institutions such as the New Zealand Ministry of Health then suggest that despite having no record of them, there are several examples of the virus being isolated and cultured in a laboratory setting, and then direct you to studies that have done no such thing in their methodology sections. They are always without fail tissue culture proxy experiments, in which the word isolation has become detached from its understood meaning, and it has never been demonstrated that any particle of the millions found in these smorgasbord tissue cultures imaged or imagined, has the properties of a disease-causing virus. In any case, it is a distraction from the wider issue exposed by Christine's Freedom of Information requests, which is that particles claimed to be viruses can never be found in human subjects. As Dr. Stefan Lanka points out, density gradient centrifugation is the scientifically required standard technique for the demonstration of the existence of a virus, despite the fact that this method is described in all microbiology manuals as the virus isolation technique. It is actually never applied in experiments meant to demonstrate the existence of any pathogenic viruses. The defense of virology's methodologies is obviously attempted by its own promoters, including New Zealand government and state-funded media's favored microbiologist, Susie Wiles. Her employer, the University of Auckland, is among those institutions who have now confirmed that it has not done any work relating to the purification of any COVID-19 virus, and therefore has neither found in nor isolated from any human subject the so-called virus named SARS-CoV-2. Yet this associate professor, above an assistant and below a professor, told the whole country that
the world is on fire, and I don't think people realize that. So we just need to, what our government is doing is it's listening to the experts and it's trying to keep us safe. And so we might actually get off this relatively unscathed if we all behave as they're asking us to behave. Yes. And we might end up having to put some more strict measures in the months or so to come.
She was then ordained New Zealander of the Year in 2021 for helping millions globally see past the fear and complexities of the pandemic.
The world is on fire, and I don't think people realize that.
And helping to keep us safe. In her November 2020 article, Koch's Postulates, COVID, and Misinformation Rabbit Holes, Wiles alleged that the people like Christine Massey asking for evidence of the existence of SARS-CoV-2 virus responsible for COVID-19 are specifically wording their request to rule out obtaining any evidence that the virus exists. Her article quickly went off on a tangent about Koch's postulates being unsuitable for viruses, and she thus declared them as invalid in that context. Robert Koch actually stole his postulates from Friedrich Gustav Henle, and ironically, Koch never fulfilled them himself in his own experiments. But Henle's postulates were a simple four-step way to prove a microbe caused a disease or not. You simply take a sick animal or person suspected of having a new disease like COVID-19, draw their blood, sputum, or fluids suspected of having the causative agent, and you must isolate that agent only, using first maceration in fluid, then filtration through a nanomesh screen like a strainer, to get out all other material in the fluid larger than the suspected causative agent. Then spin that in a density gradient centrifuge so all particles are in their own density band layers. Then extract only the layer with the suspected causative agent with a nanopipette, which virologists don't do. Then, if required, or should we say if even possible, create more alleged viral particles in a test tube and repeat the purification process with density gradient centrifugation, which virologists don't do. Then take that pure culture and inject, spray up the nose with an atomizer, or drop into the eyes of a healthy animal or person, and it must cause the exact same illness and symptoms as the animal or person you took the fluids from, which virologists don't do. Then you have to draw fluids from that animal and repeat the exact same procedures from the first two steps to see if the suspected causative agent is now in that person's fluid as well, proving transmission, which virologists can't do without cheating steps 1 through 3. But then you have to do a control experiment, taking the same fluids from a healthy animal or person and repeating the exact same steps and procedures to both check yourself and prove that you don't find the exact same results in your culture because you either cheated and didn't isolate the suspected causative agent from the thousands of other things in a fluid sample, or you cheated and didn't use a pure culture, and the results of the culture are really from the chemical toxins and foreign tissues you used in the culture itself, which virologists don't and won't do, because it will prove they are cheating. That's why they are so hell-bent on getting rid of Koch's postulates. It is strange that nowhere in her article did our associate professor Susie Wiles ever mention Rivers' postulates, which were purposely modified from Koch's postulates to include theoretical viruses, since Koch's were created for bacteria. It was even made much easier, because Rivers allowed for cultivation in unpure cultures in host cells, like monkey kidney tissue, dog epithelial cells, and even human lung cancer cells, plus toxic chemicals and antibiotics. Plus, injecting that rotting cell culture into an animal only had to produce a comparable disease and not the exact same symptoms or disease. But it is not clear on the meaning of comparable.
And I want to poll the audience. You could just shout out, what are the symptoms of COVID-19? What? Fever. Cough. Shortness of breath. Good. Fever, cough, and difficulty breathing, right? So what symptoms did they actually have? Weight loss and slightly bristled fur. Anyone hear news reports of bristled fur in COVID patients? Weight loss also not a symptom of COVID-19 that's reported. It's a very non-specific symptom. So once again, the experiment fails on this count, that this is not the same symptoms as the disease in question.
Perhaps Wiles didn't mention Rivers' postulates because she would have to admit that these postulates have never been fulfilled either, and that virologists have spent much of the 20th century trying to identify viruses directly from sick humans without any success. Awkwardly for Wiles, the World Health Organization stated back in 2003 that with regard to SARS-CoV-1, conclusive identification of a causative agent must meet all criteria in the so-called Koch's postulate. The additional experiments needed to fulfill these criteria for SARS-CoV-1 are currently underway at a laboratory in the Netherlands. The World Health Organization removed that article from their website in 2021, obviously because those experiments failed and people were asking questions about SARS-CoV-2. Luckily, we have the Internet Archive. Here's why they failed the WHO's requirements.
Some seem to claim that this paper that appeared in The Lancet in July 2003 established that SARS-CoV-1 could indeed be implicated as the monocausal agent. But wait a minute, what actually went on in this study involving 4 poor little monkeys? Firstly, they took a sample from a single human SARS victim and produced a cellular culture in the lab through several steps. The monkeys were then infected by squirting this mixture into their tracheas, noses and eyes. On the 2nd, 4th and 6th days, the monkeys were anaesthetized with ketamine, an anesthetic known for its hallucinogenic properties. Blood samples were taken from veins in the groin and smears were obtained from the nose, mouth, throat and anus. The monkeys were all killed for autopsies after just 6 days. 3 of the monkeys became lethargic after 2 or 3 days. On the 4th day, 2 developed temporary rashes. Only 1 monkey had any breathing difficulties, while 3 showed non-advancing tissue damage to the lungs. The lymph nodes near the trachea and the spleen were larger than normal in these 3 monkeys. The other organs all appeared normal under microscopic examination. Now, there are some glaring weaknesses with this study. Firstly, the monkeys' illnesses were not typical of SARS. Only 1 monkey had breathing issues and there was no mention of any of them having a cough or fever, which are some of the key features of SARS. And none of the monkeys became critically unwell or actually died from SARS. They were all euthanized by the researchers. Secondly, the paper claimed to identify coronavirus particles in monkey tissue on electron microscopy, but many different virus-sized particles are part of the cell culture, so without particle purification, it's impossible to know exactly what they were. Now, there seems to have been a lot of confusion about isolation and purification. Purification means that we've got rid of the host cell material and culture medium and just have the alleged virions by themselves. Virions are the complete viral particle, meaning it includes its shell and everything inside. So if it's not purified, then there will be other viral-sized particles in the material, which may appear in the electron micrograph images and may also contribute other genetic fragments. Thirdly, injecting 4 mL of culture solution directly into a small animal's trachea is a way to cause a lung reaction. Given that these little primates weigh around 6 or 7 kgs, it would be the equivalent of injecting, say, 50 mL or almost 2 fluid ounces of culture solution into a human subject. Introducing that amount of material directly into the trachea has the potential to flow into the lungs and cause reactive changes and even potentially a pneumonitis. On this note, it is also unclear what conclusions you can draw from injecting viral cultures directly into an animal's upper respiratory system and then a few days later finding it in the lower respiratory system. Forgive me for being a stickler for the rules, but overall this monkey business has not helped confirm Koch's postulates as they claimed.
Wiles also appeared to be at odds with Na Zhu et al., one of the teams that first claimed to have discovered SARS-CoV-2, because they conceded that, although our study does not fulfill Koch's postulates, our analyses provide evidence implicating but not proving 2019 nCoV, later SARS-CoV-2, in the Wuhan outbreak. Additional evidence to confirm the etiologic significance of 2019 nCoV in the Wuhan outbreak include animal, monkey experiments to provide evidence of pathogenicity. Whether different virologists want to entertain the validity of Koch's postulates or not, it is simply another distraction, as the postulates require the physical isolation of a microbe, rather than assertions that one exists, through means such as computer in silico simulations, or imaging vesicles of unknown biological function, like these identical alleged SARS-CoV-2 particles found in the lungs of both COVID PCR positive and negative patients, or these alleged SARS-CoV-2 particles found in the kidneys of a COVID negative patient, or claiming that unpurified biological toxic soups given to or injected into animals contain viruses. Wiles admits that when virologists want to isolate a virus from a sample, meaning take that virus alone and apart from the sample itself, they instead take the whole sample, or some part of it, and add it to some cells, usually ones that are relatively easy to grow in the lab. That is not isolation. Sputum, fluid, and lung tissue samples have all sorts of particles in them, including lung tissue, nasal tissue, or swab cheek or throat tissue itself, which all have their own RNA and DNA, bacteria and microbes in the sample, which all have their own RNA and DNA, inhaled plant pollen, which has its own RNA and DNA, inhaled bug and food debris in the nose, mouth, and lungs, which all have their own RNA and DNA, particulate matter from air pollution like dust, dirt, soot, smoke, lead, etc. And then they add that to some cells like monkey kidney tissue, which has its own RNA and DNA, and then add the blood from an aborted cow fetus for nutrients, which has its own RNA and DNA, and profess they can grow, find, and isolate a virus out of all of that, but can't find it in the original sample. Wiles says, and then we look to see if the cells die and or if there are any virus particles released into the liquid nutrient bath the cells are growing in. In other words, viruses can't be grown in a pure culture as described by Koch's postulates because they need a cell to grow in. Yet the virus particle would need to get into that cell in the first place somehow, grow, multiply, and shoot out of the cell by themselves, without the cell, to fly around the atmosphere and get into other people's cells to cause infection. Isn't that why we're told to wear N95 masks? To stop the actual tiny flying virus particles themselves from getting in and not flying host cells full of virus particles?
What's more, an N95 mask is actually really good at filtering both the largest and smallest small particles. It's medium-sized small particles that are hardest for it to block.
Isn't that the point of taking a sputum, fluid, or tissue sample in the first place? Because they should be teeming with infectious replicated virus particles. If coughing or spitting that same tiny amount of fluid can allegedly infect someone else? Despite never isolating one to check, virologists postulate that virus particles are protein and lipid shells around a capsid containing DNA or RNA, and anywhere from 50 to 140 nanometers in diameter. What biologists call exosomes, or extracellular vesicles, for example, are also claimed to be a protein shell around messenger RNA and are also 30 to 150 nanometers in diameter, exactly the same as alleged viruses. And Wiles admits, yes, exosomes, more commonly referred to as vesicles, are small particles that can be separated from samples by centrifugation. But the important distinction here is that they are bits of our cells that bud off and generally require huge volumes of material to isolate them from. The same process just can't be applied to a small sample of cells from someone's nasal swab. Yet, molecular chemists find and isolate molecules smaller than just 1 nanometer, much smaller than 150 nanometer exosome vesicles or alleged virus particles every single day without problem, and do all sorts of things with them. And IBM unveiled in 2021 the world's first 2 nanometer chip technology with 2 nanometer nanosheets, which they are easily able to manufacture, find, isolate, and even attach to other nanostrips that are only 12 to 44 nanometers wide and only 75 nanometers long, which they can clearly manufacture, find, and isolate in order to attach the smaller 2 nanometer nanosheets onto them. Yet, virologists can't find this in a large sputum sample? Wiles admitted it can be done for same-sized exosomes and vesicles with a large enough sample, but then a virologist 20 times her senior said this.
We asked a guy who was 20 years, senior virologist at Wuhan Institute of Virology, 20 years head of Yale Virology Laboratory. Can you find SARS-CoV-2 in any fluid of any person you say has COVID? He said no. Why not? Because there's not enough virus to find. Okay, we asked him, what about if you put 10 people with supposed COVID and you mix their bronchial fluid together? Is there enough? No. 100 people? No. 1,000 people? No, not enough to see. 10,000 people, you mix their fluid together. Would you then have enough virus to see? And he said no, and then he wouldn't answer anymore.
Yet this minuscule amount of fluid is supposed to contain enough virus particles to make a person dangerously sick from up to 6 feet away? As Dr. Bailey points out, it is unclear if Susie Wiles is implying that isolating a virus is established by taking the entire cocktail sputum soup of various materials from a sick person, or just some part of that sputum soup, or adding either of those to foreign tissues and fluids in a test tube that have nothing to do with that sick person, or is it just seeing some foreign tissue die in a nutrient-starved and poisoned test tube? Or is it the release of the dead tissue debris claimed to be virus particles in that test tube or tissue culture, or all or some combination of these elements? In fact, nothing Susie Wiles describes even requires the existence of viruses. as Dr. Stefan Lanka's control experiments finally proved, repeating their same foreign tissue culture procedures and getting the exact same cytopathic results with no added sputum sample at all. It is all a game of deception, whether realized or not, and it is time we all see through how it is being played against us. The claims that SARS-CoV-2 has been shown to exist through this methodology are all scientifically invalid, as none of the experiments were performed with valid scientific controls. Virologists are supposed to be studying alleged viruses that spread between, attack, affect, and harm human tissues, not monkeys. So why are they using green monkey kidney tissue in these cell cultures? In fact, the issue extends beyond just SARS-CoV-2. With the exception of bacteriophages, which are very real and not viruses, every virus asserted to exist relies on similar pseudoscience. The history of virology reveals that the types of cells eventually selected for these experiments have been those that have a propensity to break down with the claim of virus-induced cytopathic effects or cell destruction. For example, Viro E6 monkey cells from the kidneys of African green monkeys have long been favored by virologists, supposedly due to their suitability to host many viruses, but suspiciously also because the aneuploid kidney line with an irregular number of chromosomes is more susceptible to toxic insults from the additional ingredients they add, such as the kidney nephrotoxic antibiotics and antifungals added to their culture mix.
So this is how they do the procedure, very different from what I described. They call this virus isolation. It's really a tissue culture experiment. And they take the fluid from the sick person that they suspect has a viral disease. They filter it very coarsely. Sometimes they do a basic centrifugation. This is not purification. It's just to remove debris that forms a pellet, but it doesn't even remove all of the cells. And then they do a tissue culture, but they vary the conditions of the cells. They usually use foreign cells, like monkey kidney cells. They may use other foreign cells. Occasionally, they use human cells. They also use fetal cells on occasion. But they don't continue the normal culture conditions. Instead, they reduce the nutrition to 10%, generally speaking, sometimes lower of the normal amount. Then they use a modified nutrition, which has is less nutrient dense. And then what they do is they add fetal calf serum, and that, because it's from a fetal animal, it has some factors in there that improve the survival of the cells in the culture and also is a source of protein nutrition. But of course, it's a foreign material, blood product. So if there are such a thing as viral particles, it would certainly be a source of contamination. It's also been shown in the literature to be a source of contamination for any downstream genetic testing. So if they were to do a PCR test, for example, on the culture fluid, the results would be contaminated by the fetal bovine or fetal calf serum. There are papers that looked at that that basically say that the genetic testing would be worthless if you add this constituent. Then they add the antibiotics, and they do these at higher than normal concentrations, usually 2 or 3 times the normal concentration. They often pick antibiotics with nephrotoxicity like gentamicin, aminoglycosides, amphotericin, and streptomycin when they're using kidney cells. And under these conditions, they observe the culture and they find what they call cytopathic effects. As you can see, there is some changes in the morphology. There are these plaques that form, and this is a sign of the cells are in the dying process. And this is what's put forth as the proof that there was a virus way back in the original sample of the lung fluid, even though it's not known what else was in that lung fluid. They sometimes do some molecular testing to rule out certain known bacteria and viruses, but there are flaws because many of those tests are not validated. But they really don't do a compositional analysis and say what's everything that's in there, and of course there's no control experiment. So it's really erroneous to interpret it this way. So back to the issue of control experiments. Were any control experiments ever performed? So this is a paper by John Enders, and this is the original paper where this method was developed, the tissue culture isolation experiments. And Enders won the Nobel Prize related to the polio vaccine for developing this procedure, but it was developed as a manufacturing procedure of vaccines. So they used to think you could only, quote unquote, culture viruses using the type of cells that it infects. So for polio, using spinal cord or nerve cells. As some of you may know, those are very difficult to culture in a laboratory. So he had the idea to use some kind of fetal cells, which were much easier to grow. They're, practically immortal in the laboratory. And he mixed in, the toxic ground up spinal cord from people who died of polio in with the fetal cell culture, and was able to use that to manufacture the polio vaccine. And I think he helped either Salk or Sabin, I don't remember for sure. But nonetheless, in this paper, which is about measles, he used the same procedure with a different cell line, but it wasn't necessarily for the purpose of manufacturing a vaccine. In this case, or at least it wasn't published, it was for investigation. They talked about propagation in the tissue cultures. But in this experiment where they did, essentially what I just described, except they added a few other things like milk to the sample, they did the procedure without a source of measles and got the same exact results, the cytopathic effects. And they, wrote in here that that could either be from another virus from the kidney cells, unknown, of course, always blame a virus, or it could be from unknown factors, right? But never considered that it was the tissue culture conditions themselves that could have been the cause, even though that is the purpose of doing a control experiment. And in the conclusion of this paper, by the way, it doesn't say that it proves the existence of a virus, the cytopathic effects. And it also says specifically that what they found in vitro does not necessarily correlate with anything that happens in the patient with measles. It was only after this publication and after the Nobel Prize was awarded that virologists began adapting this procedure suddenly to prove the existence of viruses rather than to study viruses or manufacture vaccines.
In plain English, alleged viruses should break down the cells from healthy human adults in a test tube as well. But since their experiments failed to do any such thing, they switched to more susceptible, chromosome unstable kidney tissue from green monkeys, or alveolar A549 cells from human lung cells sick and dying with cancer, already breaking down naturally by cytopathic effect instead. Dr. Charles Dauger, the electron microscopist for Dr. Luc Montagnier, credited for discovering the alleged HIV virus particle, said, quote, "We have never seen HIV virus particles in the purified virus gradient. What we have seen all the time was cellular debris, or the aftermath of already broken down cells, not HIV virus particles." It doesn't matter how cellular tissue dies, whether you starve and poison it via necrosis, or it dies naturally from apoptosis, or you just squish it to death between two glass plates like this. When you look at the aftermath under an electron microscope, you get this dead cellular debris that looks like what are alleged to be virus particles. The ones on the left made by squishing a cell to death between two glass plates. The ones on the right made by virologists starving and poisoning a cell to death in a test tube or petri dish, and then poisoning and destroying it even further to view under an electron microscope. Or as they say in Thailand, same but different. In a June 2020 paper entitled Severe Acute Respiratory Syndrome Coronavirus 2 from Patient with Coronavirus Disease, United States, when this group attempted to culture SARS-CoV-2, they had no desired result with human adenocarcinoma cells, A549, meaning human lung cancer cells. No luck with human liver cells, HUH 7.0, or with human embryonic kidney cells, HEK-293T, or even with a big brown bat kidney cell line, EFK3B. But then miraculously declared they had a viral isolate, following the observation of cytopathic effects in green monkey Viro E6 kidney cells. They admitted from the wells in which cytopathic effect, meaning the killing, breakdown, and deterioration of cells into small particles, were observed, meaning after adding the exact same lung sputum sample from a single patient alleged to have SARS-CoV-2 to 5 different types of cell tissue lines, then starving and poisoning those tissues, they still only found that procedure only killed the cells of green monkey kidney tissue and not the rest. Hey, 1 out of 5 ain't bad. Of course, if you only had a 1 out of 5 chance of your parachute opening, would you trust the parachute science enough to jump out of an airplane? As is typical, there seemed to be no sense of irony for them that the purported human respiratory virus cannot be shown to infect the relevant cell type, let alone the relevant human species, and their experiments were once again invalidated by the absence of appropriate control cultures. Why not at least use African green monkey lung cells instead of kidney cells, if this is an alleged respiratory virus and not a kidney virus? And if you're not an African green monkey yourself, and even their own experiments can't break down decrepit alveolar A549 human lung cells already dying of cancer in a test tube, why would you believe your own healthy lung cells in your own nutrient-dense body would be more susceptible to such an alleged but unproven theoretical virus? Nonsense. And since they refuse to scientifically control their own cell culture experiments and debunk themselves, they instead control our societal culture itself by changing the societally agreed language definition of the word isolation to propagate their germ theory fraud. And that's why isolation matters. A further and major embarrassment for virology is that alleged viral particles that have been successfully purified after an unpurified cell culture beginning have not been shown to be replication competent, meaning able to multiply in a new host cell, or disease-causing by themselves. In other words, what particles have been at least partially physically quote-unquote isolated, along with the other junk in the test tube, never directly from a living being as they should be found, but only after tissue culture contamination, can only be said to be extracellular vesicles. Nothing more, nothing less. In May 2020, a publication appeared in the journal Viruses that claimed, nowadays, it is an almost impossible mission to separate extracellular vesicles and viruses from each other by means of canonical vesicle isolation methods, such as differential ultracentrifugation, because they are frequently co-pelleted together due to their similar dimension. Meaning, Because the particles they allege to be viruses are the exact same size, weight, shape, and density as all of the other identical cell debris particles, they say after they centrifuge them that it's impossible to take out the alleged virus particles only with a pipette, because they are in the exact same density band as all of the other junk, just like this cherry tomato is in the same layer as the dish soap. They are admitting they can't suck out the cherry tomato without the dish soap coming with it, or vice versa. Nowadays means in contrast to how they've always done this since the beginning, and it is unclear how such an observed technical change may be reconciled with biological laws. They've just now figured this obvious fact out for the first time in 60 years? It appears more likely that the virologists are distancing themselves from their own techniques in order to avoid refutation of their own false postulates. They may have to accept that the reason it is impossible to separate extracellular vesicles from viruses using differential ultracentrifugation is because their assertion that viruses are present in the sample are ill-founded, meaning their alleged viruses are extracellular garbage debris, nothing special or different. In 2017, the Perth Group pointed out in their magnum opus, HIV, a virus like no other, that in virology, while purification retains its everyday meaning, isolation is an expediential term, meaning advantageous to them, that virologists assign to data, not physical reality, they claim are proof a particular virus exists. The paper states neither virologists nor the scientific literature provides a satisfactory definition of virus isolation. HIV expert Jay Levy defines virus isolation as a sample of a virus from a defined source. White and Fenner as the ability to identify a totally unforeseen virus or even discover an entirely new agent. Montagnier and Weiss as propagating them, viruses, in a cell culture. The 2013 6th edition of Field's Virology defines virus isolation as viruses can be isolated from an infected host by harvesting excreted or secreted material, blood or tissue, and testing for induction of the original symptoms in the identical host, or induction of some abnormal pathology in a substitute host or in a cell culture. Once the presence of a virus has been established, it is often desirable to prepare a genetically pure clone. It goes without saying that if virus isolation is to take a sample of a virus from a defined source, or propagating viruses in cells in culture, one must first have proof that the virus exists in a defined source or in cells in culture. Neither is virus isolation induction of some abnormal pathology, or once the presence of a virus has been established.
There's no universally accepted definition for these terms, strain, variant, and isolate, in the virology community. So how can we expect that everyone else gets them right? What happens is you're trained in someone's laboratory and you hear them say things and you associate a meaning with them, and that's what you do. And they may or may not be right. Now, despite this looseness of definitions, for example, there's no one place where you could go and find the definition for variant, for example, or isolate, or strain. I looked in several textbooks. In fact, not even my textbook, Principles of Virology, co-authored with 4 other virologists. Not even there will you find a definition of these terms. I think we need to put it in the next edition, though. Let's go through and try and define them and put them in some kind of context with the current outbreak. Let's start with isolate. What is an isolate? An isolate is a virus that we have isolated from an infected host, and we have propagated that in culture.
You cannot define the word isolate with the word isolation or isolated as its definition or meaning. Defining something with its own name is called recursion, known as a homunculus logical fallacy. Just like you do not define the word red as something that is red in color. A blind person or colorblind person would have no idea what you are talking about, or a child who hasn't learned the names of colors yet. So you say, red is a color whose hue resembles that of blood or of the ruby, or is that of the long wave extreme of the visible electromagnetic spectrum. To a child, you point to the red blocks only and say that is red, not those other colors. You do not point to a starved and poisoned tissue culture with tens of millions of pieces of broken down genetic material from a dozen different sources and say that is an isolated virus, any more than you would think this is a pile of isolated red blocks. Virology invented the hypothesis or theory of viruses, so whatever method it employs in an attempt to prove their existence, it must satisfy that definition. At the heart of the matter is a simple concept, and we need to see evidence that alleged disease-causing particles cause new particles that are clones of the former. Claiming that detected proteins and nucleic acids are of a specific viral origin and not just normal broken down components of a tissue culture is not possible unless the alleged viral particles have been truly isolated by purification and shown to have these key biological characteristics. As outlined by the Perth Group in HIV, a virus like no other, purification is necessary to prove the existence of viruses for several reasons, including the following. Viruses allegedly replicate only in living cells. Since cells and viruses are composed of the same biochemical constituents, separation of particles from cellular material is essential for defining which nucleic acid and proteins belong to the virus particles. Also, to prove the particles are infectious. In other words, it is particles, not other factors, that are responsible for the production of new particles. This requires purification of both sets of particles. Also, to demonstrate their biological and pathological effects, and to obtain antigens, proteins, and nucleic acids for use in antibody and genomic tests, respectively.
Like you were alluding to, everything's, flipped the other way with Snopes, right? It's the same thing here. If you actually go to the studies and the information, the way in which they do the lab information, the studies, and they gather their data and they make those claims is all based on artifacts that they're manufacturing in the laboratory process, and is not what happens in the physical body, in the living body. And this has been the major con that they've done to our medicine and to the knowledge base, is that they're not watching and looking at the living blood and how things operate. They are always removing particles, removing tissues, altering them through their methods, whether it's petri dishes, antibiotics, other types of cells like kidney cells from the monkey, or fetal cells, or their methods of growing the so-called virus. And then they're adding, coloration and adding UV light and other light when they're, viewing things under microscopes. And they're actually manufacturing an illusion with artifact production. And now they're taking artifacts and trying to study them further and make claims about them. To me, it's absolute insanity, because they've never done the control studies and the proper methods to then corroborate it back with the story that happens in the living body. They're assuming that what happens in the petri dish or under the microscope is what happens inside of us. That is a huge glaring error of science. It's not scientific method structured, and it's not validated. They don't ever validate those things.
You've probably heard the term virion, meaning a theoretical complete viral particle consisting of RNA or DNA surrounded by a protein shell and constituting the infective form of a virus. Though no proof of one exists, other than a wild guess as to what these destroyed tissue culture particle images may possibly be made of or possibly do, they theorized them to be possibly like this, just like a pink-horned unicorn horse theoretically might look something like this, or maybe even something like this. Although less common, virologists will sometimes obfuscate the meaning of purification as well. Purification means the removal of contaminants from something, meaning getting rid of everything else in the tissue culture and leaving only the alleged virus particles, or the process of extracting something from a substance, meaning taking out the alleged virus particles only and leaving all the other tissue culture garbage there. On May 23, 2022, Belgian professor of virology Mark van Ranst claimed that with regards to SARS-CoV-2, in another article, they have further purified the virus by ultracentrifugation in beta-cyclodextrin. Van Ranst was referring to a 2008 paper that described large-scale preparation of UV inactivated SARS coronavirus virions, which related to the purported SARS-CoV-1 virus. However, this paper simply outlines a protocol claiming to purify virions, and there is no part of the paper that demonstrated the existence of any replication-competent particle. All that was shown were some low-quality images purporting to show infected Viro E6 cells. With regards to the check of purified virions following centrifugation, no images were provided, but the claim was made anyway that the concentration of purified virions is determined by BCA or bisynchroninic acid assay with bovine serum albumin as a standard, which is a joke because all a BCA or bisynchroninic acid assay can do is detect the total concentration of a protein in a solution. It cannot determine whether that protein is from a virion or just plain old regular proteins abundant in tissue cultures. In other words, it can tell you there are exactly 5-hoofed, 4-legged animals with tails and manes here, but not if they're unicorns or just regular horses. In an October 2013 paper in the journal Nature, we find a paper entitled Isolation and Characterization of a Bat SARS-like Coronavirus that Uses the ACE2 Receptor. We are given this ridiculous image of completely different vesicle particles in size, shape, and color. If I were an alleged scientific expert on cloning, what would you think if I claimed that these people were all replication-competent identical clones of each other? Yet, the convenient variation in particle size is apparently because coronaviruses usually have a diameter, excluding projections, of between 80 and 120 nanometers, although in extreme cases, the diameter can vary between 60 and 220 nanometers. Because the definition of identical clones is clearly as loose as the definition of isolation to virologists. But you see, Mark van Ranst cites a fancy-worded fluff paper, sodium dodecyl sulfate, polyacrylamide gel electrophoresis of proteins, which sounds so scientific that you'll never bother to question it. By really just claiming that confirming the existence of a virion or virus particle using gel electrophoresis, which just simply separates different proteins from each other by their molecular mass, is a better way, is as big of a joke as using bisynchronicic acid assay, because gel electrophoresis too cannot provide any evidence that a protein even belongs to an alleged virus or so-called virion. Only that it is a different mass and density weighted protein than other proteins with different molecular masses. but at least his way can tell a big yellow horse from a little brown horse, but still not if it's a unique yellow unicorn or just a plain old yellow horse. But Mark Van Ranst is a virology expert, so we should trust him, right?
To tell you about my experiences being the crisis manager, the flu commissioner for Belgium, and highlighting the communication aspects there. And then you have one opportunity to do it right. I mean, day one is so important. In day one, you start your communication with the press, with the people, and you have to do it right. I mean, you have to go for one voice, one message. In Belgium, they chose to appoint a non-politician to do that. I mean, I have no party affiliations, and that makes things a little bit, at that time at least, a little bit easier, because you're not attacked politically, majority, minority. That doesn't come into play, and that was a huge advantage. The second advantage is that you can play in Brussels the complete naive guy and get a lot more done than you would otherwise be able to do. You have to be omnipresent that first day or the first days, so that you attract the media attention. You make an agreement with them that you will tell them all, and if they call, you will pick up the phone. When you do that, then you can profit from these early days to get complete carpet coverage of the field, and they're not going to search for alternative voices there. And if you do that, that makes things a lot easier. And then you have to say, okay, well, we will have H1N1 deaths. Of course, that would be unavoidable. I used there Sir Donaldson's quote, where he said that in the UK, by the peak of the epidemic, 40 people would die per day at the end of the summer. So 62 at that time million people in the UK, 40 deaths a day. I worked it out for Belgium. That would be 7 deaths a day at the peak of the epidemic. I used that in the media. 7 Belgian flu deaths per day at the peak of the epidemic would be realistic. That is true in every year, even interpandemically. That is very, very conservative. However, talking about fatalities is important, because when you say that, people say, wow, what do you mean? People die because of influenza? That is true in every year, even interpandemically. That is very, very conservative. However, talking about fatalities is important, because when you say that, people say, wow, what do you mean? People die because of influenza? And that was a necessary step to take. And then, of course, a couple of days later, you had the first H1N1 death in the country, and the scene was set and it was already talked about.
Van Ranst was not the only virologist making claims about purifying viruses. In response to an email inquiry, Dr. Marissa Grossages from the Robert Koch Institute responded that we purified SARS particles by density gradient. However, just, meaning only, from the cell culture-derived virus, as you wrote. The challenge with purifying SARS from patient samples is that you won't get a visible band. Meaning, according to her, if you take the sputum from a person who is allegedly sick from millions or billions of SARS replicating viruses in their lungs, and hence in their lung sputum, and centrifuge that, they can't find a density band consisting of any of these alleged SARS virions anywhere in the test tube. But they can magically find particles they claim to be SARS virions after their tissue cultures are starved and poisoned and break down into 1 million pieces, or should we say rather, conveniently find them. When pressed for further details about how or if the experiments were even controlled, she responded, "We are not allowed to share any protocols with a private person. I can only refer to our publications, where infection experiments are described in detail." Unfortunately, no such promised details were given in any of the experimental studies she provided. SARS-CoV-2 remains nothing more than a hypothetical computer construct assembled from genetic fragments of unproven provenance. It's as if outside of a chemically controlled and manufactured petri dish, viruses are nothing more than a figment of the imagination. As Mark Bailey points out, There has never been a physically isolated, i.e. purified particle shown to be responsible for the production of identical particles or a particle shown to be the cause of pathological effects in any human or in an experimental animal model. In other words, they've never observed any submicroscopic particles they label viruses do any of the things they claim they do. Thus, the declaration by virologists such as Mark Van Ranst, along with the WHO and its adherents, that an infectious particle termed SARS-CoV-2 is causing a disease pandemic is shown to be patent scientific and intellectual fraud. So what is virology? When startled, the mythical Uslam bird will take off and fly around in ever-decreasing circles until it somehow manages to fly up its own ass, disappearing completely, which adds to its rarity. It is hard to know exactly what to call virology, but it is not science. The current practitioners are engaging in some form of algorithmic or statistical speculation added to circular reasoning and confirmation bias, searching for, interpreting, and recalling information in a way that supports their theory, or what we commonly call cherry-picking data. With a complete absence of what should be the corresponding process of refutation that lies at the heart of the scientific method, i.e., trying to debunk their own theory with valid control experiments in the lab and actual observations of isolated particles causing contagion and illness in the real world, not just cartoons and Hollywood movies. While the abandonment of the scientific method may be accidental or unnoticed by lower-level virologists and lab technicians, there are almost certainly conspiratorial motivations at higher levels of the globalist hierarchy. For example, the WHO, the Center for Disease Control, and the United Kingdom's Health Security Agency are all parties to virology's deceptive practices. However, the anti-scientific practices are also replicated in most other countries, whether this relates to claims of virus isolation and the wholesale misapplication of the polymerase chain reaction, or PCR test, for clinical diagnostics, or a failure to disclose the crucial control details involved in virus culture and genome creation. Pseudoscience consists of statements, beliefs, or practices that claim to be both scientific and factual, but are incompatible with the scientific method. In other words, pseudoscience cannot be proven, tested, or repeated. It's just a baseless theory, like the pseudoscience germ theory of disease. Science, and even solid scientific theory, can be proven, tested, repeated, and most importantly, falsified, meaning solid scientific theory can be demonstrated to be false. You can have a pseudoscientific theory, like flying unicorns actually fly backwards instead of forwards, but it's impossible to either disprove or falsify that theory or prove that theory true, because there's no such thing as flying unicorns in the first place. Refutation is at the heart of genuinely testing a legitimate scientific theory through the scientific method. As Karl Popper wrote in his book, The Open Society and Its Enemies, about scientific theory and method, so it is, I hold, the possibility of overthrowing it, the scientific theory, or its falsifiability, that constitutes the possibility of testing it, and therefore the scientific character of a theory, and the fact that all tests of a theory are attempted falsifications of predictions derived with its help, furnishes the clue to the scientific method. The view of the scientific method is corroborated by the history of science, which shows that scientific theories are often overthrown by experiments, and that the overthrow of theories is indeed the vehicle of real scientific process. The contention that science is circular cannot be upheld. But for science to be true and not just pseudoscience, it must be logical and reasonable. Yet circular reasoning is a logical fallacy, meaning illogical and unreasonable, in which the reasoner begins with what they are trying to end with. The components of a circular argument often seem logically valid, which is why the pseudo-virologists use circular reasoning, because if the premises are true, the conclusion must be true. But a premise simply means an assumption or theory. As Karl Popper said, your control experiments are there to try and debunk your own premise, assumption, theory. Real science does not fix and rig experiments through dodgy, unscientific methods to make their baseless claims appear to be true, and then not bother to run any control experiments to debunk or falsify their own premise. But I'll tell you who does do that: con men. This woman is sick with flu symptoms. I wonder if there are submicroscopic particles called viruses inside of her, causing these symptoms. Virologists will tell us. Step 1. Collect her lung sputum containing the virus particles. That is circular reasoning. You are supposed to try and disprove through falsification experiments your premise that those particles are even there in the first place, not baselessly assume for no reason whatsoever other than your virology school-taught belief that they are there before you even start. Circular reasoning is illogical, unreasonable, and thus unscientific. Dr. Bailey was correct when he stated, "It is thus a reasonable question to ask. Has virology ever been a scientific pursuit?" With regard to the scientific method, the virologists create unfalsifiable hypotheses by setting up paradigms where any number of observations, whether it be illness or alleged test results, can be attributed to their viruses. The observations are passed off as proof of virus existence in the manner of a circular loop of reasoning that no longer requires the demonstrable existence of a virus. Any claims of reproducibility, for example, in the form of a PCR process or purported viral genome, are simply more circuits of the same loop. Historically, virology has been characterized by a lack of valid control experiments, and none of its foundational claims have been established through proper exercise of the scientific method. Again, falsifiability. They should take sputum from a completely healthy person and assume there is no virus. At least make sure that person tests virus negative with the PCR test, even though it doesn't matter. Then poison and starve that healthy sputum on the exact same cell culture using the exact same chemicals for the exact same period of time. Let it all break down into 56 million tiny genetic fragments, and then tell their computer to arrange those fragments like a puzzle until they match the fictional computer in silico genome for that alleged virus. If they can do that, which virologist Dr. Stefan Lanka did successfully do himself, it proves it is their procedure causing the cytopathic effect breakdown of tissue in their culture and not an alleged virus. The first alleged virus to be discovered was the tobacco mosaic virus, which looks remarkably just like rotting lettuce in your cold fridge. And one of the proofs for this is said to be contained in Dmitry Ivanovsky's 1903 treatise about the mosaic disease of the tobacco plant. However, it is patently clear that Ivanovsky's described experiments lack any valid control comparisons and were thus, surprise, surprise, unscientific and inconclusive.
Here we've got the conflation of sick plants with a virus, where we're jumping from A to Z here without any of the proof that's required in the middle. So one thing, we got sick of hearing this too, that Ivanovsky is the father of modern virology, and he's got this great paper, late 1800s, 1900, we're talking now, and that this proves that this was the first ever documentation of a virus. But if you read that paper, Ivanovsky doesn't claim that he's found a virus in the modern conception, and I think that's where the public get confused, and even people who are doctors and scientists in the system get confused as well, because they don't actually read the source material. Ivanovsky simply observed that some plants got sick and others didn't. He even notes that it seems to be related to where they're planted, i.e. the terrain, the environment. And we all know that you can't plant lemon trees in Greenland. We don't say that, goodness, we don't say every single citrus tree we've planted in Greenland has come down with some sort of virus, because it just won't take, So if you actually read Ivanovsky's paper, he talks about this contagious fluid that he's run through filters. But again, the methodologies he used were insufficient, because he's simply taking material from one sick plant and injecting it into a healthy one and watching it get sick and saying, well, there must be some sort of agent, transmissible agent. And yes, there is an agent, but it's not contagious or transmissible, because grounding it up and injecting it into another plant doesn't replicate what happens in nature. So Ivanovsky, there's nothing there. And it's clear, he even says in those experiments that he planted a sick plant, which was alleged to have the tobacco mosaic virus, but we should just say it's got tobacco mosaic disease is more accurate. And he planted healthy plants right next to it, like, within centimeters. And hey presto, that plant didn't get sick.
Yeah, so where's the virus?
Yeah, his own experiments basically document that it didn't transmit, and unless he did these abnormal kind of grind up and inject methods, which went completely out of control.
It is tempting to forgive the early pioneers of virology that their uncontrolled and unscientific methodologies were simply typical practices for that era. However, germ theory critic Claude Bernard offered the following insight into the importance of control experiments when adhering to the scientific method decades earlier in 1865. If indeed we characterize experiments by a variation or disturbance brought into a phenomenon, it is only insofar as we imply that the disturbance must be compared with the normal state. As experiments indeed are only judgments, they necessarily require comparison between two things, and the intentional or active element in an experiment is really the comparison which the mind intends to make. Bernard was advising the need to have a valid control or some suitable comparison to ensure it was only the new experimental element that was causing an outcome. In plain English, if you suspect a dress makes you look a bit fat, you need to try on a bunch of other dresses to see if you look skinny in those, to make sure it's not just the fat underneath the dress that makes you look fat. It is possible Bernard was the only scientist of his time in virology's pursuit of invisible enemies who actually understood the scientific method, but not probable. Moving forward to another early claimed virus discovery, the textbook Retroviruses informs us that, in 1911, Peyton Rouse at the Rockefeller Institute in New York reported the cell-free transmission of a sarcoma, meaning a tumor arising from the connective tissue, in chickens. The virus, isolated by Rouse, bears the name of its discoverer, Rouse sarcoma virus. However, a review of Rouse's paper, A Sarcoma of the Fowl, Transmissible by an Agent Separable from the Tumor Cells, reveals that he did not claim to isolate anything, let alone anything that met the definition of a virus. His methodology involved grinding up chicken tumor material, filtering it, and injecting it directly into other chickens, with the observation that some of them would also develop tumors. He reported that the control experiments consisted of injecting unfiltered tumor material into chickens, which tended to result in much larger tumors. Rouse postulated, meaning took a wild guess, at the presence of a causative ultramicroscopic organism, but conceded that an agency of another sort is not out of the question. Indeed, the experiment failed to provide any evidence of an infectious and replicating particle. It simply showed that diseased tissue introduced by an unnatural and invasive route into another animal could cause it to exhibit a similar disease process. Would you be shocked if someone ground up this rotting sarcoma tumor, injected it into your connective tissue, and you developed a tumor at the injection site? Would you say you caught a sarcoma virus there? This debunks William Guy's 1925 work, because finding a tumor is not proof that a virus caused the tumor. It simply demonstrated tumor formation following injection of diseased tissue. Further, it was determined in 1927 that sarcoma of the fowl could be induced by the injection of dilute arsenious acid in foreign embryonic pulp. The carcinogenic effects were also replicated following the equivalent bacteriological filtration that Roos performed, and the disease was shown to arise from the foreign tissue, not from the host tissues. Carell's summary stated, "We have discovered the presence of an important phenomenon, the formation by the tissue under the influence of a nonspecific chemical substance of a principle which resembles a virus, acts in a specific fashion on the cells of a certain type, and reproduces itself indefinitely in the presence of these cells." The viral hypothesis should have been thrown out right there, but instead, half a century later, the establishment kept it alive and rewarded Roos with a Nobel Prize in 1966 for his discovery of tumor-inducing viruses. Then in 1954, when John Enders and Thomas Peebles claimed they had propagated the measles virus in human and monkey kidney cells, no further tolerance should have been extended to virology's unscientific experiments. Enders and Peebles added throat washings and blood to their cell cultures, and on observing cytopathic effects, or the dying and breaking down of cells in their test tubes, concluded that the in vitro or in scientific test tube appearances might be associated with the virus of measles. Might is used to indicate a condition or state contrary to fact, or used to indicate a possibility or probability that is even weaker than may. They did at least warn that cytopathic effects, which superficially resemble those resulting from infection by the measles agents, may possibly be induced by other viral agents present in the monkey kidney tissue itself or by unknown factors, but then went on to inappropriately conclude that this group of agents is composed of representatives of the viral species responsible for measles. They did do one lazy control experiment where they admitted a second agent was obtained from an uninoculated culture of monkey kidney cells, meaning they didn't put any material from a person with measles in there, and that the cytopathic changes it induced in the unstained preparations could not be distinguished with confidence from the viruses isolated from measles. That actually invalidated the evidence for their conclusion, but ideally, several control experiments should have been done, some with no human-derived samples added, some with human-derived samples added but from people who are well, and some with human-derived samples from unwell subjects but said not to have measles clinically or some other alleged viral condition, just sick people. That is how you do real science, and this is how you do not. The virologists, however, have continued to repeat the uncontrolled methodology of Enders and to this day claim that such cytopathic effects are incontestable evidence of viruses. Dr. Stefan Lanka has documented the history of these unscientific practices and in 2021 demonstrated that cytopathic effects could be induced in cell cultures by the laboratory process itself. The results of Lanka's experiments are depicted here. proving you can easily and repeatedly break down healthy tissue in a lab causing cytopathic effect without adding any material sample from a sick person, simply by following the cell culture procedure techniques virologists adopted from Enders and Peebles. In many virology publications, a control or mock-infected experiment is mentioned, but the details of such experiments are conspicuous by their absence. Much like our mock unicorn control here. A Northwestern University, Illinois webpage states that mock-infected means a control used in infection experiments. 2 specimens are used, one that is infected with the virus vector of an interest, and the other is treated the same way except without the virus. The definition is already flawed, as terms such as virus and infected have been introduced and thus presumed to exist before actually being established. In June 2022, in response to an Official Information Act request concerning the paper, characterization of the first SARS-CoV-2 isolates from Aotearoa, New Zealand, as part of a rapid response to the COVID-19 pandemic, the University of Otago stated, the paper published by Professor Quinones Mateu and colleagues was a descriptive paper. This means there was no hypothesis to prove or disprove. In 2008, the journal Infection and Immunity featured a guest commentary titled Descriptive Science that explained why descriptive research by itself is seldom conclusive and may simply serve as a starting point to orientate further investigations. The authors pointed out that microbiology and immunology are now experimental sciences, and consequently investigators can go beyond simply describing observations to formulate hypotheses and then perform experiments to validate or refute them. As this essay outlines, the virology establishment will not divulge or carry out these required experiments, seemingly in order not to refute itself. It intentionally limits itself to ongoing opportunistic fishing expeditions backed by confirmation bias, thus disqualifying itself from the scientific method due to its inconsistency with the hypothesis-driven and falsifiable approach described earlier by Popper. If we're supposed to trust the science, should they not at least be beholden to actually use the scientific method and not hypothetical descriptive science? Dr. Mark Bailey has previously written in a postscript derived from A.F. Chalmers' book, What Is This Thing Called Science?, that one of the pivotal issues with virology was that it invented itself as a field before establishing if viruses actually existed. Literally the cart coming before the horse, and it has been trying to justify itself since its inception. But if pink unicorns have never been proven to exist, why invent unicornology? And don't you think unicornologists might have some incentive to cheat to prove they do exist in order to keep those big paychecks coming in? As Dr. Mark Bailey wrote in Warning Signs You've Been Tricked by Virologists, in this instance, a virus particle was not observed first and subsequently viral theory and pathology developed. Scientists of the mid and late 19th century were preoccupied with the identification of imagined contagious pathogenic entities. The observations of the naive inductionist did not identify a virus a priori and then set about studying its properties and characteristics. The extant presupposition, meaning just conjecture or presumption, of the time was that a very small germ particle existed that may explain contagion. What came thereafter arose to fulfill the presuppositional premise. We shouldn't even call it the germ theory of disease, because even a scientific theory demands evidence that has repeatedly been tested and corroborated in accordance with the scientific method. And so it is clear that viruses have never even reached the stage of a theory. According to the science itself, they remain mere speculation. And thus, virology's lack of controls means it is not a scientific pursuit. OIA requests have revealed that New Zealand's Institute of Environmental Science and Research, who have claimed isolation and genomic sequencing of the SARS-CoV-2 particle in the Antipodes, are also guilty of failing to perform any valid controls. In the tradition of enders, they have not paused to check whether the cytopathic effects they witnessed or genomes they assembled via computer simulations could also be created in valid control comparisons. That is, by performing experiments with other human-derived specimens from both well subjects and unwell subjects who are said not to have the alleged disease COVID-19. Instead, ESR described their insufficient negative control in which the flask undergoes the same conditions as the flask used for viral culture. However, we use infection media only. Again, the whole point of control experiments is to test whether an independent variable can be identified, not change multiple variables and assume that the results of the experiment can only be explained by one thing, in this case, an imagined virus. The central conductor in these anti-scientific pursuits is the World Health Organization. It is very telling that in their 94-page genomic sequencing of SARS-CoV-2 document, there is a mere 4 sentences discussing control samples. However, neither of these negative or positive controls are sufficient to validate the genomes that the virologists are producing through these techniques, because they can only serve to calibrate the pipeline. And as become apparent, the WHO cannot point to one valid positive control experiment. Yet, on February 11, 2020, they named the new disease they had invented, COVID-19, with the associated claim that it was caused by a novel coronavirus. They have provided the green light for anyone around the world to find SARS-CoV-2 in their backyards without the need for valid control experiments. Yet, there is a clear necessity for comparative controls where similar patient sputum samples without the alleged virus, like these 2 healthy and sick people who tested negative, are processed in the exact same way, so that only one variable is being tested. Comparing the results of a sputum sample alleged to contain the virus from a positive PCR or antigen test with one of the negative controls described by the WHO's document above cannot validate the process, as the latter samples of buffer or water do not contain the sputum genetic muck soup that is part of the former sample. With all of the failures to culture postulated viruses, modern virology now favors direct metagenomics of crude samples. The NIH tells us metagenomics is the study of the structure and function of entire nucleotide sequences isolated and analyzed from all the organisms in a bulk sample. That is not isolation. We're no longer interested if there really are virus particles in this tissue culture soup, or if there really are green blocks in this pile, because we know blue and yellow mixed together make green, and there are blue and yellow blocks in this bulk sample. So if our computer Castle block genome template requires green blocks that aren't in the sample, we'll just sequence all the blue and yellow blocks in this pile together and say this bulk sample technically has green blocks in it where the blue and yellow primers meet. Could you make this castle out of these blocks? Virologists can. That's exactly how metagenomic computer software can assemble 56 million random short 150 long ACGT nucleotide sequences from a tissue culture rich with multiple broken down genetic sources into any 30,000 sequence long genome you tell it to, be it an alleged virus genome or a banana genome. And that doesn't even include the probability algorithms programmed into the software when it can't fit certain pieces together precisely and needs to take a wild guess, a fictional computer-created genomic band-aid, if you will. Is there any recipe you couldn't make if you had 56 million ingredients at your disposal? They sometimes call this shotgun sequencing, and subsequent artificial computer assembly of these genetic fragments allows virologists to create new in silico viruses, meaning viruses existing in a fictional computer model construct only, out of thin air. This software invention then provides other virus hunters with pre-designed PCR primer panels, so they can also discover the same meaningless sequences and claim it is the same virus. The artist no longer has to manually mix yellow and blue to make green. The computer's software just assumes green is there if it detects any yellow and blue, and then puts green in the sequence automatically, whether it is really there or not, like claiming the Ukrainian flag is technically green, if you grind it up into 56 million micro pieces and re-stitch it all together in alternating micro colors. ESR were involved in a publication in which they proclaimed the discovery of SARS-CoV-2 in 9 subjects through this methodology. But when asked to provide all details of the control group that was used when comparing the results of sequencing, they refused and instead sent links to SARS-CoV-2 sequencing protocols. If ESR were using these protocols, then we can see that they are endorsing insufficient controls that are described as a negative control of nuclease-free water, while an optional positive control can also be included, which may be a synthetic RNA constructs or high-titer clinical sample which can be diluted. Once again, these types of controls can only serve as pipeline calibration techniques, not the validation or the clinical significance of any genomes they assemble. On July 19th, in response to an OIA request, they stated that ESR has not performed any experiments to scientifically prove the existence of SARS-CoV-2 virus and can therefore not provide you with any records. On the 17th of August 2022, in response to another request, they admitted that ESR has not performed any experiments to scientifically prove that the SARS-CoV-2 virus causes COVID-19 and can therefore not provide you with any records. Nobody else has performed these required scientific experiments either. So they have to rely on animal abuse and antibody studies. With the inability to demonstrate the physical isolation of a disease-causing particle that meets the definition of a virus, the virologists have engaged in animal experiments to convince the uninitiated that such pathogenic particles exist. And like clueless monkeys studying other monkeys, these animal experiments also lack valid control experiments. An illustrative example of this monkey business was the paper Comparative Pathogenesis of COVID-19, MERS, and SARS in a Non-Human Primate Model, published in May 2020 by a team that included none other than Christian Drosten and Ron Fauchier. The nonsense of what was published in Science can be summarized as follows. The 8 cynomolgus macaque monkeys in the experiments were inoculated with SARS-CoV-2 under anesthesia via a combination of intratracheal, 4.5 milliliters, and intranasal, 0.25 milliliters per nostril routes. Pouring fluids down the nose and throat of a sleeping monkey is not a natural exposure route, and 4.5 milliliters poured into a small 3 to 5 kilo monkey's lungs is equivalent to pouring around 1/3 cup of foreign biological material into a human's lungs while they're asleep. The inoculum poured into their lungs and nose was made from SARS-CoV-2 isolate beta-CoV Munich BAVPAT1 2020, obtained from a clinical case in Germany from a German traveler returning from China. The virus was propagated on Viro E6 cells, unstable green monkey kidney tissue cells, in Optimum, Glutamax, and then supplemented with kidney nephrotoxins, penicillin, and streptomycin, not to mention the kidney-dissolving enzyme trypsin. They have asserted that they have a viral isolate, yet neither they nor their supplier have demonstrated the existence of a virus in the sample. All that can be said is that the sample contains foreign biological material from the human-derived clinical sample and monkey kidney cells, in addition to cellular breakdown products and 2 antibiotics. Even after being waterboarded in the nose and lungs with this noxious fluid, none of the older macaques showed the severe symptoms that humans do. In fact, no overt clinical signs were observed in any of the infected animals, except for a thin serous nasal discharge in one aged animal on day 14 post-inoculation. No significant weight loss was observed in any of the animals during the study. In other words, despite the direct entry into the lungs with what they claimed was the SARS-CoV-2 virus, it didn't make any of the monkeys noticeably sick. But yet it allegedly killed nearly 7 million people? By day 14 post-inoculation, or being waterboarded with toxic muck, all remaining animals seroconverted, meaning they developed antibodies in their blood serum, allegedly as a result of infection or immunization, as revealed by the presence of SARS-CoV-2 specific antibodies against the virus S1 domain and nucleocapsid proteins in the sera. Yet these S1 and nucleocapsid proteins have never been shown to be viral in origin, regardless of whether they induce the detection of other proteins termed antibodies in a host. The virologists once again employ circular reasoning to claim that the detection of an antibody proves the existence of a virus because the antibody is declared to be specific to only the alleged virus. Despite antibody scientists themselves assuring us that these so-called antibody proteins bind to all sorts of non-specific things or nothing at all, depending on the environment, the state of the proteins, whether acidic or basic, oxidized or reduced. Every scientist who has carried out such experiments or studied them knows this, yet every virologist conveniently seems blindly or willfully ignorant of this simple fact. In fact, we see this same circular reasoning with vaccines. How do I know the COVID-19 vaccine works? We can reliably prove it with an antibody test. Yeah, but an antibody test confirmed that I'm already immune to COVID-19. You can't reliably prove that with an antibody test. As a measure of so-called virus shedding, nasal, throat, and sticking Q-tips up the rectum of the monkeys were analyzed for virus by reverse transcription quantitative polymerase chain reaction, or RT-PCR. But there was no virus shedding. There was simply detection of the same sequences that had been recently poured into the monkey's respiratory tracts. It's like pouring a third cup of milk into a monkey's lungs and 2 weeks later saying, "Oh my God, we found milk in there." Of course you did. You put it there, and you are specifically only looking for short genetic milk sequences with your PCR test. Yet these foreign nucleic acid sequences, unsurprisingly, disappeared from the monkeys' bodies over the next few days through natural clearance mechanisms, despite not being given any vaccines or antiviral medication. So why are you taking them? SARS-CoV-2 RNA was only detected in a rectal swab from 1 out of the 8 animals on day 14, and no viral RNA was detected in whole blood at any time in any monkey throughout the study. No viral RNA was detected in the blood, aside from our moving nervous system fluids, which should be able to spread alleged respiratory virus particles from our lung tissue up to our brain and out to all the other connected organ of the body they allegedly never also spread to. And seeing as our digestive system doesn't connect our lungs to our rectum because it bypasses the lungs and starts at the stomach, the blood circulatory system is the only other way alleged respiratory virus particles could possibly get from our lungs to our rectum without infecting every other organ tissue in between on the way. It's the only other moving body transport system that connect the two. Again, this indicates that they were only finding the same introduced genetic material in the same places it had been introduced. The single positive rectal swab may have been a false positive, or the monkey had swallowed some of the introduced biological material. In not one case could they demonstrate that the postulated virus had any invasive characteristics. 4 of the monkeys were murdered and autopsied 4 days after inoculation with the foreign biological soup. 2 out of the 4 were reported as having small foci of consolidation in their lungs, or what you might call pneumonia from damaging their lungs. And the authors stated that the main histological lesion in the consolidated pulmonary tissues of both the young and aged animals involved the alveoli and bronchioles and consisted of areas with acute or more advanced DAD, diffuse alveolar damage. The histological features, meaning taking a wild guess what you're looking at under a microscope, were asserted to be characteristic of SARS-CoV-2, even though the same study says the other organs in these 2 macaques, as well as the respiratory tracts and the other organs of the other 2 animals, were normal. And that is because the top 3 slides across in their paper, A, B, and C, are consistent with pneumonitis, caused by pouring a liquid containing foreign biological material directly into the monkey's trachea while it was anesthetized. The histological appearance changes in the bottom 3 slides across, D, E, and F, simply depict inflammatory tissue cells, such as macrophages and neutrophils, as would be expected in such inflicted pneumonitis. No control experiments were performed, pouring a 3rd cup of any other fluid down their trachea to see if the exact same pneumonia-causing cell tissue damage occurred. The alveoli are the end point of the respiratory system on the lungs and allegedly exchange oxygen and carbon dioxide in the bloodstream, sort of like the little green balls on the end of broccoli. And the bigger cells on the outer surface of those little balls that do this gas exchange are called type 1 pneumocytes. The paper claims SARS-CoV-2 antigen expression was detected in moderate numbers of type 1 pneumocytes and a few type 2 pneumocytes within foci of diffuse alveolar damage. The problem is, this was claimed through an immunohistochemistry staining technique that was based on a rabbit polyclonal antibody against SARS-CoV nucleoprotein. Why is that a problem? Because the supplier of this product, Sinobiological, admits IHC, FCM, IF, IP et al. Applications haven't been validated. Antibodies applications haven't been validated with corresponding virus-positive samples. And this tonic will make your hair grow back, even though it's never once been proven to do so and the guy selling it to you is still bald. In any case, this example can be used to expose the wider fallacy regarding antibodies as any evidence of viruses. Sinobiological states that the antibodies were the result of injecting their SARS-CoV nucleocapsid protein, his tag product, into rabbits. This nucleocapsid protein was in turn produced from a DNA sequence encoding the SARS-CoV isolate TOR2 nucleoprotein. by use of a product on the market with testing in progress. We'll get into the so-called TOR2 protein in Part 2, but in summary, outside of a computer-simulated hypothetical in silico model, no protein has ever been shown to come from a virus, including the nucleocapsid protein in this case. It was baselessly asserted that they injected viral proteins into animals instead of just other regular foreign proteins, and in response, the animals produced other proteins that are claimed to be antibodies. A virus was neither shown to exist nor required to exist for this sort of pointless exercise. Again, we are told a specific viral protein like SARS-CoV-2 will make the body create a specific antibody protein that will only bind to that alleged SARS-2 virus protein and nothing else. Yet, the COVID-19 vaccine generated HIV antibody proteins in 100% of the volunteers injected with the University of Queensland candidate vaccine. Looks like there's a major problem with the HIV hypothesis. If you can have so-called disease-defining HIV antibodies without any possible HIV virus to trigger the body to create them. Getting back to the study at hand, it did not follow the scientific method as it lacked controls. Not only were there no control experiments, but they didn't even demonstrate animal-to-animal disease transmission from the waterboarded monkeys allegedly infected to healthy monkeys. If these viruses are so airborne and infectious, why do they have to inject monkeys or pour fluids down their nose and throats? Why not simply aerosolize a sample into the animal cages so they inhale it? Once again, such experiments are avoided in order for the virologists not to refute themselves with regard to claims of contagion involving the imagined particles. Which brings us to the virus quantity paradox. We are led to believe that inside a host such as a human, the viral particles are produced in such great numbers that they can rupture the very cells containing them, while at the same time, paradoxically, they are present in such tiny amounts that virologists say they can't be seen in any patient specimens. Apparently, with regard to the alleged SARS-CoV-2 particle, it has somehow been magically calculated that one sneeze of a COVID-19 patient contains 200 million viruses. However, if we obtain a physically larger sample directly from a subject's nose or lungs, equivalent to hundreds of sneezes, there are precisely 0 to be found, rather than billions, as claimed. So virologists pull the magic rabbit out of their hat by claiming they are invisible while grown in your lung sputum and can only become visible once grown in their magic tissue cultures.
Now, here's the first question. Since the viral theory is I breathe in this virus, it uses my lungs as a culture, right? And so then it grows, and then it's teeming with viruses that get spewed out in the world. Why can't you find it in your lungs? It's a culture.
Oh, your point is if you do a direct electron microscopy of the fluid, you won't see this.
Never.
Okay, yeah, I see.
Never. Not once. We asked a guy who was 20 years, senior virologist at Wuhan Institute of Virology, 20 years head of Yale Virology Laboratory. Can you find SARS-CoV-2 in any fluid of any person you say has COVID? He said no. Why not? Because there's not enough virus to find. Okay, we asked him, what about if you put 10 people with supposed COVID and you mix their bronchial fluid together? Is there enough? No. 100 people? No. 1,000 people? No, not enough to see. 10,000 people, you mix their fluid together. Would you then have enough virus to see? And he said no, and then he wouldn't answer anymore. So,
but he claims what we see on the EM, on the electron microscopy slides, are in fact virus particles. The little bleb
what
things.
He says is you have to culture them. There is no virus to see in your lungs,
right,
or in your snot.
Enough that they'll admit
anywhere.
Yeah.
Both Dr. Mark Bailey and Dr. John Bevan Smith also destroyed this virological nonsense in their paper, The COVID-19 Fraud and War on Humanity. So we are supposed to believe that the human respiratory tract, which is lined with the alleged perfect host cells, does not produce enough viruses for them to be seen. But a test tube experiment involving tissues from a different species and cell type does? Pfizer suggests to the layperson that the immune system reacts to the injury of these bodily cells by revving up, but did not cite any scientific evidence for this imaginative claim. In fact, aside from poisoning, nobody can prove there's anything else to even be immune from. The fourth edition of Medical Microbiology has offered up 4 hypothetical pathogenic mechanisms. Hypothetical means an assumption but not fact that can be tested by further investigation, and pathogenic means mechanisms that cause a diseased or morbid condition that may result in cell damage and death, or they may not. But again, even if these speculative measures were at play, it would require enormous numbers of cells to be affected to produce symptoms, just like it would take enormous amounts of institutions worldwide to keep this fraud going. But unlike the very real institutional viruses of this world, we can never find these virus particles in our own cells or tissues, because virology has a very real habit and unique way of diverting attention away from such aspects that raise doubts about its phantasmal model of disease. End of part 1 and the end of virology.